The presence of a pathogen or toxin can be detected using known antibodies or by detecting specific antibodies in a patient. These types of tests are called immunoassays. Enzyme-linked immunosorbent assay, ELISA. ELISAs use antibodies labeled with enzymes to detect antigen-antibody reactions. Enzymes are used because of their safety, ease of detection, and relatively low cost. The most commonly used enzyme is horseradish peroxidase. In the direct ELISA technique, known antibodies are used to identify an antigen. A known antibody is absorbed to the wells in a microtiter plate. A patient's sample is added to the wells. Any of the antigen in the test sample will be bound to the antibody. A second antibody that reacts with the antigen is then added. This antibody is linked to an enzyme. The reaction between the antigen and antibody is made visible by the addition of the substrate for the enzyme. We're going to look for salmonella in eggs. Mark each egg with a code to make them different. Each egg must be homogenized. Transfer a working sample of each egg to a microtube. A standard microtiter plate contains 96 wells. You will use one row for your experiment. Label the wells in your row 1 through 6. Place 50 microliters of coating buffer into wells 1 through 6 of your microtiter plate. Place 50 microliters anti-salmonella enterica in wells 1 through 6. Cover the plate with tape and leave the plate undisturbed for 30 minutes to allow the antibodies to be absorbed to the well surfaces. Invert the plate and with a quick shake, remove the liquid into disinfectant. Fill the wells with washing buffer and remove the buffer into disinfectant. Pipette 50 microliters of the negative control in well 1. Pipette 50 microliters of the positive control, a heat-killed salmonella, into well 2. Pipette 50 microliters of the first egg sample into well 3. Repeat to transfer each egg sample to its well. Cover the plate with tape. Incubate the plate for 30 minutes at 35 degrees Celsius to let the antibody react with salmonella antigens. Add 50 microliters of washing buffer to each well and then remove the liquid and discard in disinfectant. Add 50 microliters of peroxidase-labeled antibody to each well. Cover and incubate the plates at 35 degrees Celsius for 30 minutes. Add 50 microliters peroxidase substrate to each well. Cover and incubate for 15 minutes at room temperature. Observe the wells for color. We're using tetramethylbenzidine, TMB, as the substrate. The horseradish peroxidase enzyme will make a yellow compound from the TMB. The negative control is blue. The positive control is yellow. Egg samples 3 and 5 are positive for salmonella. Dispose of the microtiter plate in the proper disposal area.